Jackie, right there.
It's hard to understand that biomass is not an important trade-off. You may get, I'm surprised actually you don't get a relationship between harvest index and biomass. Maybe it's a population of different genotypes, I don't know. But if you look at just genotypes of same harvest index, I think you're going to get, there is a strong hypothesis that until unless you increase biomass, you probably won't increase the potential. And then it comes to another important issue is growth duration, which I think is missing very much in this genotype analysis we did.
Yes, those are both very good points. I would agree with both of those that you know, the fact that we have these data sets that don't show a relationship with biomass. I mean, one interpretation of that lack of relationship is that natural selection has really worked really hard on that, and that there's not that much room for improvement in biomass. You know, if you're only comparing materials that are locally adapted and so on. But I'm not going to lean too hard on that, because in the both of those studies that I showed actually were grown at a single plant density. And so a lot of these, you know, leaf angle and stem number and other traits that we haven't thought of, I think do have the potential to increase biomass, but they only have that potential at high density. So we need to explore that further. And I forget what your second point was. Oh yes, no, I think that's a really important point, and I sort of tried to raise that point after John's seminar, that, you know, the time when net increase in dry weight stops increasing, you may not want to, that may not be the time to harvest. You may want to harvest, you know, earlier than that, when you're, you know, to maximize production per day, not production per growth cycle, and then squeeze in another harvest in the year. So, yeah, very good point, John. I think you get to defend yourself first.
No, it's fascinating to talk. There's so many things that could spend the whole afternoon talking about it. One of the things I wondered, when you showed the Kasman-Dubik picture, it was 60 years, I think, of leaf scoring, and it was a curve sloping upwards, which made it seem as if it was a long time before leaf angle had an impact. But I thought they only started really selecting for it around about 1970. And if we were to look at the rate of increase in yield from 70 on, it's quite rapid. That would be my impression.
So you're saying there was a deliberate selection for leaf angle but only after 1970?
Well, I think starting around about 1970, because I was involved when I was in my PhD with forage grasses, and there was this great debate about, do you select for angle or do you select for leaf photosynthesis? And I can remember going back to the literature, and there wasn't much going on prior to that, because there weren't the instruments around to measure light distribution. You know, the solarimeters were the first crude attempt at that. The other point, I guess, sorry.
Certainly the case that the golden age of talking about leaf angle was that period. They say in their paper that plant breeders didn't have that as a criterion for mating. So the literature was out there, but you know, plant breeders have lots of things to think about and they can't use them all. So I don't know whether their statement is correct, but I bet it is because Dubik was one of the authors.
It went up, but I thought it accelerated down to the 70s, and that probably reflected the fact that they were being positive in selecting for it. You know, so progress increased because they were after that particular target. That was my impression. Sorry.
It's a bit of a chicken and egg question, maybe, because the other thing that has happened, that you don't know which one is the cause or effect, is that since the early 1960s, the plant density in commercial corn cropping in the US has essentially doubled. They used to grow it at 20,000 plants per acre or less, and now the standard density is above 32,000 per acre already. And of course, as an interest from seed companies, we have more of that than you can sell more seed. But it has led to essentially a plant type that breeders were selecting for because they were forced to grow under higher densities, and became a single ear plant type with erect leaves. Whether that was the cause or the consequence, I'm not sure, but it's a major change, and it's probably, in my view, actually, the most important factor contributing to rising corn yields in the US.
Yeah, so when you start breeding at high density, you're going to get erect leaves as a plyotropic effect of second for yield under those conditions.
Sorry, just one other general point. That you were making this point about specificity in Rubisco. It was 20 million years ago, wasn't it, that CO2 was around about 100, 120, 150 parts per million. And we've had 20 million years since then, and not much has happened with Rubisco, I think. Is Rubisco an intractable case, almost, for change in plants, and if so, why? Sorry, that may be too difficult in general question.
I'm sorry. What was CO2 in 1500s?
I think it was probably in the region of 300, 290 parts per million. It's 20 million years ago that it was down at the hundreds, because that's when Rowan Sage says the driver for C4 evolution started to go, or the scenario for selection for C4. And if you look at CO2 change across time, it goes from about 3,000 down to about 200, and then it kind of drifts along to about 300 parts per million, and then starts to climb up in the Industrial Revolution, but fairly slowly at first. So I think that 100 parts per million is probably a long time ago, Sorry, I don't need to be taking that time yet. I just wondered whether Rubisco was a difficult case. I mean, evolution had a lot of time to work on it, and is it a molecule that you can't do a lot with? It evolves under zero oxygen. It's pretty good at what it does, but you can't do a lot to change it. I know you mentioned cyanobacteria, but again, I wonder whether that was a kind of an aquatic environment which the gas ratios would be different.
Yeah, it'd be interesting to look at all the species in that trade-off graph and look at how many mutational steps are there between them. That would give some indication. I mean, certainly, the graph where I had the two way up there, that would be my explanation for that, is that probably that's such a big genetic change that even though it breaks the trade-off, it's too difficult. But I think some of those on the lower graph were also cyanobacteria, so I don't know.
I don't.
Yeah, maybe my understanding of the concept of idiotype is not quite correct. I was just wondering, instead of drawing a dichotomy between selecting for idiotype and selecting for yield, why don't we go for yield and then base the idiotype on what we're getting yielded?
Yes, and then what? Go breed for yield, then identify the resulting idiotypes, and then do you go back and use those idiotypes in subsequent breeding?
Well, in a way, yes, because is there really a dichotomy between the two? I mean, if we are going just for the yield and we are going for the idiotype, there must be some major differences between the two.
Well, ideally, the definition of the idiotype is those traits that result in high yield. So there isn't that kind of an intrinsic competition. It's a matter of how do we get the plants that have the high yield, which will then have the idiotypic traits. But those traits won't necessarily be what we thought they were. That's the tricky part. So the real question is how good are we at figuring out what kinds of traits will lead to high yield? If we're really good at that, then it may be faster and easier to select for those traits, and that may be the fastest and easiest way to get to high yield. If we're not so good at it, then it may be better to select for high yield.
So in terms of at least drought tolerance, it seems it has been much more useful selecting for yield under drought than actually looking for all the secondary traits that were associated with drought tolerance.
Yeah, let me give you another. I mean, there was some discussion years ago about nutrient use efficiency, nitrogen use efficiency, which ended up sort of being defined in practice as you have high nitrogen use efficiency if you have more yield relative to the amount of nitrogen in the plant. And I think that would be a case where that would be a really bad idiotype trait, whereas high yield under low soil fertility would be good. And it maybe that's true for. I'm not going to disagree with you. I think that this is sort of an open question, and based on the kind of results you get, we may start to understand what the key traits are for drought tolerance, and maybe some of those traits will be things that we can identify, either through molecular markers, obviously, but also perhaps through traits like stomata per square centimeter or who knows what, that then might be easier to measure accurately than it is easier to measure yield accurately. And if so, that could be useful. But I'm really not pushing for abandoning breeding for yield by any means.
Thanks. The trade-off between compatibility of mixtures and yielding ability in pure stands obviously is applicable when you're comparing mixtures with pure stands. And in the context of a breeding program, that means comparing early generations with late generations. If you measure the yield of an F6 or F7, which is a pure stand, you're measuring yield ability. If you measure the yield of an F2, you're measuring mainly compatibility. And that was established quite a while ago. I think you'll find that, the yield of the plot on F2 is primarily driven by the most competitive plants in it.
Oh, yeah.
So early generation selection for yield is known to be not very effective. And you'll find most breeding programs start their yield testing only later on.
Yeah.
Precisely because of this reason, that it's known not to be effective in early generations. And idiotype breeding will work in early generations if your idiotype traits aren't similarly sensitive to the contrast between mixtures and pure stands. But I think that's the concept that idiotype breeding will help you make earlier generation selection, provided you're careful with your choice of idiotype trait. I'm afraid I haven't read that 2011 paper, so I haven't seen how this applies in there. You might be able to tell me.
Yeah, no, I think that's a good point. So certainly, for example, disease resistance mostly is going to be similar in individual plant versus a group, though not completely. So that seems like something that would be relatively safe to assess at an early stage. But I agree that most of the sort of fundamental yield potential determining things, if you want to assess them at an early stage, it has to be done very carefully.
So maybe a bit of history on the Yuan paper, 2011. So that goes back to a discussion that we started in 2007. When I came back to Erie in 2007 and became program leader of Program 2, I was surprised that nobody was talking about yield potential anymore. That word had almost disappeared out of the vocabulary in Erie. And basically nobody was actively working on it in a systematic manner. And so we started the process of discussions, probably for a year and a half at least. And so environmental work was the breeder, of course, involved in this, and then on the physiology side was Sharping Peng primarily, but a number of other people, and some of them maybe in the room here. And discussion went around this question, okay, what have we done wrong before and failed in increasing plant type and all of these things, And we couldn't quite resolve all of that. And then the discussion went around next, saying, well, what do we need to do differently? What are the traits that we should look at? And so it came to a new idiotype concept. Tanguy Lafarge was heavily involved in that too as a physiologist. And then the question becomes now, how can we test some of these assumptions in there of these traits? That's how the study came about, where Xiaoping at some point suggested, well, let's just do it empirically in the field. I'm going to go out with my group to select plants based on individual idiotype traits we think are right from a physiology point of view. And Parminder is going to go out on a separate day with his group to select plants or entries based on what he thinks is right as a breeder. That's how it was done.
Yeah.
As you have pointed out, it wasn't completely conclusive in some ways, because maybe we've missed a few critical traits that we should have looked at. But I feel that there is some value in continuing that kind of exercise, and particularly looking also at some of these traits that we haven't looked at. One of them is that single stem concept. The single stem. Rice breeders, at least those at Erie here, have always bred for tillering ability because we plant our nurseries at 20 by 20 centimeters, mostly. And now we do some direct seeding here and there, but I don't think we've really consistently looked at this question of different idiotype sets, depending on how we actually establish the crop. I think that there are still gaps in this concept. I remember a year and a half ago, I'm not sure who was along, we had a temporary rice consortium meeting in Krasnodar in Russia, and we were looking at some of their breeding materials there, and those looked like Donald's wheat types. They were single stem individual plants, basically, bred for, and even the planting would almost look like wheat ears sticking straight up. But it was rice. Wow, Japonica. We never had this kind of plant type in indicas, and we had a discussion of this. But I think, just to summarize from my angle, I think these types of studies, as you have pointed out, is quite interesting to at least continue in a more systematic manner, because I think there are lessons to learn also addressing the question of what do you select for at what stage in the breeding program.
I'll be quick one. I just want to throw my thought in this question of Ajay. I think idiotype-based selection, of course, logical. If you're sure, when you said that, you're sure of those idiotypes. But the problem we face, especially in case of rice, is plasticity, which Amelia mentioned. And that's where you can be very wrong, and you may not reach anywhere. And that's why if you look at historically, breeders have done it for years, and then physiologists have explained it afterwards, post-mortem. And so I know jury is out there, which approach is the best one, but I think considering so much plasticity we have and rice changes, if you change tillers, then it changes some other thing, in spite of numbers changes. So probably yield-based selection is probably the best way to go.
I think to really make progress on models, we have to start thinking about plasticity as part of the phenotype. What kind of plasticity has been favored by past natural selection, and what kind of plasticity is going to be most beneficial in agriculture?
Can I just come back there? I just want to make a point while I can see the stem first of all. At one stage, we got involved in the calculations that how many stems you have, all that sort of stuff, which came out of the new plant type work. In the new plant type, we got 11.5 tons in the new plant type and here, because we were supporting both of them with I suppose you'd call nets. No difference in yield, a fair difference in the number of tillers per unit ground area. And we were looking at unicorns and wanted, in fact, to go and have a look at a unicorn. They were breeding, they were selecting one, they were genetically modifying some of the rice types in China. They got it, and it turned out to be a disaster because the gene controlling tillering same one was controlling branching. And so they ended up with this miserable plant that, there was absolutely hopeless. I mean, the whole project got jumped out of that. I think the important thing, one of the important things is that you do have to concentrate on light interception. And so leaf area is the first step. I need a leaf area of this amount, and I can divide it up by as many stems as I like in the ground area, which is going to influence leaf size. If on these units you've got something that can give you the grain, number of grains, or the weight of grain that you require, you're fine. But I think that almost should be the starting point. As opposed to the other way around, numbers of stems per ground area.
But early in the season, in the growth of the crop, leaf area index 2 on a multi-stem cultivar at 20 centimeter spacing is going to have less light perception than leaf area index 2 on a fewer stem per plant and closer spacing.
That's been long known. And if you do the calculations, what you find is a crossover. As soon as the canopy starts to close, after about 20 or 30 days or so, then the erect one starts to take off. It's much more productive. In the calculations you do of canopy photosynthesis, it's the photosynthesis that you're getting in the region of flowering on that becomes important, not the first 2 weeks. And distributing the light over that large area as you come up to that important phase is important, and that's what erect will give you. I mean, ideally, you might say what you want is a plant, and I think Shelby told me that they got these in China once upon a time. Leaves that are horizontal initially, and they get more erect as the canopy closes. I didn't know whether to believe him or not, or whether he was pulling my leg.
Yeah, this is basically what they're breeding for now in China, where the goal is to have the upper 3 leaves erect, or 4, or actually the leaf angle of each of the upper leaves getting steeper and steeper as you go to the younger leaves. The older leaves are more flat.
But if what you have in the lower part of the canopy is sunflex, then a sunfleck on a horizontal leaf is small, and a sunfleck on a tilted leaf is illuminating a larger, the same sunfleck illuminates a larger area. So I'm not sure you necessarily want horizontal leaf at the bottom.
Well, I think we're soon going to have 3D printers who can print us any idiotype we want, and then we can put it into the field or expose it to the light for various experiments, and then let's go from there. Good, that's a good discussion. We continue tomorrow morning, 9 o'clock, is the next topic. Thanks for coming.